Hello friends, welcome back to Smriti Notes for Biotechnology. So this video is the continuation of the previous video that was on food technology, part B. So in the previous video of part A, food technology, we studied about the temporary methods. Now in this video, we'll be studying about the permanent methods. Before I start with the first topic, if you're not able to understand any of the word, then do comment the page number and let me know. I'll clear the word because these notes are not today's notes, they are 4-5 years older notes. So now let's start, and do like, share and subscribe to my channel, and if you want notes for any other subject or subtopic, then do comment and let me know. The first method is sterilization or processing by heat. The products that are difficult to sterilize are increasing and are decreasing in protein. That means the complete destruction by heat of all forms of life in the product is sterilized. They are sealed that all living microorganisms are excluded. The second type of permanent method is sterilization or processing below 100 degrees Celsius. Like the fruit juices are processed at 65 to 85 degrees Celsius high temperature on heating at 100 degrees Celsius. The fruits are preserved at 100 degrees Celsius. Vegetables other than those with increasing acidity must be heated at 100 degrees Celsius to kill the microorganisms or bacteria. The second one is intermittent processing at 100 degrees Celsius. All these are part of sterilization or processing below 100 degrees. So, intermittent means the sterilization is divided into 3 to 1 hour each at 100 degrees. It is more effective and they are separated by 24 hours. It is usually suitable for vegetatives at decreasing acidity. The next is now acidification. Now acidification means acidifying vegetables and meats with lemon juice or vinegar. This is called lemon juice method. The pH should lie before 4.4. A small amount of lime juice is added to the brine in which the vegetables are canned so that after sterilization the pH remains below 4.5. Next type of permanent method is use of steam under pressure. The boiling point of water is raised if steam and water are closed in an autoclave and found at 120 degrees Celsius. Spores are killed easily. Or tomato juice are preserved by sorting at 120 degrees Celsius in a continuous flash sterilizer. Next is permanent preservation by antiseptic like sugar, salt and acetic acid. Now sugar, it is used in concentration of 70% or over, will permanently preserve food like fruit jellies. Salt, it acts both by osmosis and as microorganism poison, more effective than sugar. The next is acetic acid, microorganism poison also called. Better than salt and sugar. 2% of acetic acid prevents spoilage. Now there are other preservation methods as well, that is by chemical preservation, by drying. We'll not go through in detail about them, but we'll just have an overlook. Now, chemical preservatives are more inactive. The amount required raises as per the pH. Sulfurous acid may act as a permanent preservation for fruit products at concentration less than 0.2. We can also use benzoic acid. Now, for drying is also another method which reduces moisture content and increases the osmotic pressure, which decreases the growth of microorganism. Another permanent type of method is preservation by fermentation. Now, by fermentation there are various types. First is acetic acid fermentation, lactic fermentation. In acetic, the vinegar fermentation is done by vinegar bacteria. In lactic, now there is in detail about preservation by chemicals and by salting and drying method as well, but that is not included. Still, I've made notes on it. If you want, you can go through them and they will help you. I'll be sharing it. What you can do is you can just pause the video and then make a note of it. If you're not able to understand any of the word, then do let me know. See, there are so many methods which I have given, though they all are not mentioned, but still I made a note out of it so that if asked, you can write it easily. Now for preservation by drying as well. Now we studied it, what is drying, but not in detail. So here some advantages, type of drying like semi-drying, everything is mentioned. You can make a note of it. Mechanical dehydration, that is by direct or indirect heated. The next is preservation by salting. Various advantages and disadvantages are given for using salting as a preservation permanent method. There are 10 methods for preservation by salting. Now, if you are not able to understand any word or point, do comment and let me know. Now, this was the second part of the syllabus which we started on the preservation methods. So, if you have any other video or topic which you want, you can just write it down in the comment section and let me know. So now we have covered with this the very first module of the syllabus, that is module 1, processing and preservation technologies used in food industries by heating, drying and baking. Now you can relate that we have covered all these things in detail. So you can pause the video, make a note of it and learn them thoroughly. If you face any problem, then do comment and let me know. Now I'll be starting with the next module in the next video. So do like, share and subscribe to the channel. Thank you, and for more updates, press the bell icon.